Good afternoon. Welcome to this month's Superfund research program presentation. My name is Lex van Geen. I am the Associate Director of our Superfund program here at Columbia University. And we have one presentation. Typically we try to have two presentations. Today we have one. And the topic is the Geomicrobiology of Arsenic and Antimony, presented by Tom Kulp, who is an assistant professor in the Department of Geological Sciences and Environmental Studies at Binghamton University. Binghamton University is one of the State University of New York campuses. Tom received his PhD in 2004 from Indiana University in Bloomington, and he studied the biogeochemistry of selenium and some Cretaceous formations, really a geological angle to that. Then he was offered a postdoc to go to the U.S. Geological Survey in Menlo Park, a postdoc in NRC, the National Research Council postdoc, and was there for a number of years. He joined there the group of Ron Ormond, who's been a speaker here with us, and clearly someone who's been at the forefront of arsenic biogeochemistry for several decades. And in 2011, Tom moved to Binghamton, where he currently has his research group and teaches as well. I remember that the first time within the group, and this was even before Superfund started, the person who brought up arsenic and antimony as a comparison was actually Yan Zheng. This must have been before when we were trying to get funding from NSF at the time, and she brought up a study in Lake Fazin in France, where vertical profiles across a redox line of both arsenic and antimony had been studied by a French group. Okay. And Tom confirmed that that's indeed one of the sort of earlier studies where the two elements, for their similarities and differences have been studied. So I look forward to an update on that front from you, Tom. Thank you.
Thank you very much. And thank you very much for this invitation. I'm very happy to be here today. Yeah, how do we... Okay. Well, so I wanted to talk to you today a little bit about microbial cycling of arsenic and antimony, telling you about some of the research that I've been working on basically through my career with the USGS and then continuing to work on today. I wanted to talk about arsenic cycling in the first half of the talk, and I wanted to tell you a little bit about some studies that we've done in some more sort of extreme or unusual environmental settings, such as arsenic-contaminated soda lakes in the western United States or in arsenic-rich hot spring hydrothermal environments. And then towards the second half of the talk, I would like to talk about some more recent work that I've been doing at a contaminated mine site in Idaho. This is the Stibnite Mine, Valley County, Idaho, in the Payette National Forest. And I want to talk to you about some arsenic and antimony issues there related to mine tailings. Okay. Trying to figure out how to advance it here. Okay. Well, arsenic and antimony are both toxic metalloids, and they both co-occur in group 15 of the periodic table. And so, as such, they have some similarities in their chemistry and also in their toxicological properties. So, for example, in the case of arsenic and also antimony, these two elements can be present in 4 different redox states in the environment, but at surface conditions, the +3 and +5 oxygen ions are the most common forms, most common inorganic forms in the environment. So in the case of arsenic, we're talking about arsenite, the +3 redox state, and arsenate, the +5 redox state. And then in antimony, we have antimonite, +3, and antimonate, +5. you can see here the relative crustal abundance of the two elements. Arsenic is the more abundant element in the Earth's crust. The crustal abundance of arsenic is low. It's about 1.8 milligrams per kilogram. But there's evidence to suggest that in the geologic past, the concentration of arsenic at the surface of the Earth may have been higher by as much as several orders of magnitude. For example, the arsenic concentration in carbonaceous chondrite meteorites is up to about 0.1%. And it's thought that the reason that the crustal abundance has gone down through geologic time is because in the case of both arsenic and antimony, we're talking about chalcophilic elements that like to associate with sulfur. And so plate tectonics through time would have been moving these elements down into the mantle where they would become sequestered, and then they only kind of come back up to the crust through volcanism. So the idea is that through time, the relative abundance of arsenic and most likely antimony has probably gone down. That'll become important a little bit later for a point I'd like to make. Well, the role that microbes play in controlling the speciation and environmental behavior of arsenic is well established. And I know that probably most people in the audience today are quite familiar with the influence of microbial processes on, for example, the release of arsenic in contaminated aquifers, such as the situation in Bangladesh, for example. So microbes will catalyze various reactions that will alter the environmental form and redox state of arsenic. For example, microbes are known to catalyze the oxidation of reduced arsenic-bearing minerals, such as arsenopyrite or arsenic sulfide minerals. And they will drive the oxidation of these forms of arsenic to the soluble arsenate ion by coupling their oxidation with oxygen or, for example, other stronger oxidants such as nitrate. Arsenate, in turn, is a fairly sorptive oxyanion, especially onto the surface of iron oxides and aluminum oxides. Microbes can also catalyze the reduction of arsenate to arsenite by coupling that to the oxidation of organic carbon, for example, or to the oxidation of inorganic electron donors. In the process of reducing arsenate to arsenite, they're changing it from a more sorptive form to a less sorptive form, a more soluble form, which causes its mobilization into the water. And also microbes can indirectly release arsenic by reducing the iron oxide minerals themselves, for example, and liberating adsorbed arsenic into the water, where it then can be, drawn up from wells and cause a lot of the problems that we see in Bangladesh, for example. Okay, so why do the microbes do this? Why do the microbes transform arsenic oxyanions? And there's a couple of different reasons. This diagram shows three of the primary genetic operons that are involved in arsenate reduction or arsenite oxidation. If we start over here on the right-hand side, the RC operon, the Rs operon and the enzymatic products of those genes, such as RC and RSB, for example, are involved in arsenic detoxification mechanisms. So microorganisms that have the Rs operon are resistant to arsenic. And these genes encode for the ability to reduce arsenate to arsenite once arsenate is taken up into the cell. Arsenate gets taken up into the cell through phosphate transporters because of the similarity of the anion to the phosphate ion. So basically, phosphate transporters oftentimes can't tell the difference between arsenate and phosphate. Arsenate gets taken up into the cell. Arsenate is toxic to the cell, so the cell wants to get rid of it. And as it turns out, the easiest way for the cell to spit it back out is in the form of arsenite, the more reduced oxyanion. So the RC gene encodes for an enzyme that will reduce arsenate to arsenite. And then the RSB system is an arsenite efflux that will in turn spit the arsenite back out. And so the net effect of this is that microbes take up arsenate, arsenic 5, reduce it, and spit it back out as arsenic 3. And the Rs operon for arsenic resistance is actually a very widely distributed operon in nature. There are lots of microorganisms that possess this gene, both from arsenic-contaminated environments, but also in environments that are not contaminated with arsenic. For example, E. coli is a fairly arsenic-resistant organism, and it possesses the RC operon. Okay, so that's arsenic resistance. For the remainder of this talk, we're going to be a little bit more interested in sort of the latter two examples here. The first one is the operon ARR, which encodes for respiratory arsenate reductase. Basically, this is a mechanism of anaerobic respiration where the microbe is going to take electron donors, either from a heterotrophic organic source or from an inorganic autotrophic reductant in the environment, pass electrons from this reduced electron donor through a respiratory electron transport chain associated with the membrane. And it's going to use the arsenate, the more oxidized arsenic 5 form, as a terminal electron acceptor for respiration in the absence of oxygen. In the process, they're going to reduce arsenate to arsenite, from arsenic 5 to arsenic 3. So they're doing this as a metabolic respiratory process to obtain energy. And this is a mechanism of anaerobic respiration, sort of analogous to sulfate reduction. This is a widely distributed ability among many different groups of both gram-negative, gram-positive bacteria and archaea. Okay, so this is an anaerobic respiration process. And then over here we have arsenate oxidase, the oxidation of arsenic 3 to arsenic 5. In this case, the arsenite is going to act as an autotrophic electron donor. Again, it's going to pass the electrons through a respiratory electron transport chain, and it's going to couple the oxidation of arsenite to arsenate with the reduction of oxygen or some other stronger oxidant. And again, this is a metabolic process to obtain energy, to fix CO2. Okay, so this is autotrophic arsenite oxidation. So these processes have all been pretty well studied by now. This is a slightly outdated diagram from about 10 or 12 years ago, but it does serve to illustrate the point that the ability to reduce arsenate as a respiratory electron acceptor, as well as chemoautotrophic arsenite oxidation and heterotrophic arsenite oxidation, these different processes are widely distributed, like I was saying, between all the different groups of the bacteria. There are archaea with these abilities. And so the phylogenetic relationship suggests that this may be sort of a very ancient type of enzyme, a very ancient type of metabolic strategy in the bacteria. Okay. So when I started working with Ron Ormland's group at USGS, they were studying arsenic cycling in some very arsenic-rich soda lakes in California and Nevada. And in particular, they were looking at arsenic cycling in Mono Lake, which is what this picture is here. And this is sort of, if you don't know where this is, it's kind of just to the east of Yosemite National Park in the rain shadow of the Sierras. And this is a close basin soda lake. Water flows in from the Sierras, meltwater from the snow, but it doesn't flow out. It flows into this basin and basically it evaporates. And so you have evapoconcentration of the water here. The salinity is about 3 times that of seawater, about 80 or 90 grams per liter. It has a pH of about 9.8, and it contains about 200 micromolar dissolved arsenic. And the arsenic is primarily derived from the fact that there's lots of hydrothermal systems in this area and hydrothermal springs that are also draining into the lake, providing arsenic-rich water and also leaching arsenic from the geology of the area. And then once that arsenic flows into the lake, it also evapoconcentrates. Okay, so we get high concentrations, naturally high geogenic concentrations of arsenic here. And I'm also going to talk about this lake, Searles Lake, which is actually sort of a more evaporated end member of Mono. And this is in the Mojave Desert in California, and it's basically a salt-saturated playa. But if you chip down through this salt crust is, you know, maybe about 5 to 10 centimeters thick. And when you chip down through there, underneath you find a shallow salt-saturated brine, and then below that anoxic sediments. And so the arsenic concentrations here in Searles Lake, up to 3 millimolar. In Mono Lake, like I was saying, about 200 micromolar. Okay, here is Mono Lake, here is Searles Lake. This whole chain of lakes is basically the remnants of a Pleistocene lake chain that formed when the glaciers were retreating after the last ice age. And these are basically the remnants of what was a much larger lake system that would have filled these entire valleys with fresh water at the time. Okay, you can see also the salinities and the pHs I was mentioning here. Okay, so these are naturally arsenic-rich areas, and they make a pretty good natural lab to study arsenic cycling, as it turns out. So around the time that I joined Ron Ormland's group, they were looking at arsenic cycling in the water column of Mono Lake, and they demonstrated Mono Lake is a stratified water column due to density and thermally driven stratification. And so we're talking about aerobic water that overlies anoxic bottom water. Okay, and so at some depth in the deep part of the lake, you're going to reach an oxycline where dissolved oxygen goes down. And what we see going on with the arsenic that's present in the lake is that there's a complete biogeochemical arsenic cycle where arsenic 5 reduction to arsenic 3 is driven by these dissimilatory arsenic reducing prokaryotes. Okay, these are basically the bacteria that are going to respire arsenate as an anaerobic respiration strategy. They're going to reduce arsenate to arsenic 5 to arsenic 3, coupled to the oxidation of organic matter from the primary productivity in the lake. And then we see the reverse reaction at the oxycline, where we have the chemoautotrophic arsenite oxidizers that are going to make a living by oxidizing arsenic 3 to arsenic 5, and they're going to basically use the reducing power of arsenic 3 to fix CO2 into organic carbon. Okay, so this was where Ormland's group was basically studying when I joined their group. And in 2000, Ron estimated that up to 8 to 14% of the total primary productivity in the lake could be mineralized by arsenate respiration based on the arsenic respiration rates that they were measuring in the water column and the amount of arsenic that's present in the lake and the estimated productivity in the lake. In other words, they're saying that arsenic cycling is an important component of the overall carbon cycle in the lake. Okay, so in arsenic-rich environments, these processes can be significant in terms of things like carbon cycling. So we started to look at what was going on in the sediments of these soda lakes. And so that's where I came in. And this was an experiment I did early on when I joined Ron's group, looking at biological arsenic 5 reduction in sediment microcosms now from that salt-saturated Searles Lake sediment that I showed you, which was the salt-saturated crust, the playa. Okay, so when we set up anaerobic microcosms under a nitrogen atmosphere in the absence of air, and we're using just the sediment as an inoculum here and also as a source of some endogenous electron donors that are going to go in there with the sediment, such as organic carbon, sulfide, and things like that that are in the sediment. We're putting it into a artificial media, so we can control the composition. But basically, this was an artificial liquid media that we constructed to sort of mimic the Searles Lake water chemistry in terms of the major ions and so on, and the salt concentration. Okay, so at salt-saturated conditions now, we see that in killed control slurries, in microcosms that we establish and then autoclave, to kill them, to kill the microbes, when we give them an artificial arsenic amendment of about 1 millimolar or so, nothing happens in the kills controls, okay? The arsenic 5 remains as arsenic 5 over the course of a 25-day incubation, and we see no change from arsenic 5 to arsenic 3. But in the live conditions where they have not been autoclaved, we see reduction of arsenic 5 and at least a partial recovery of that arsenic 5 as arsenic 3. The reason we don't have 100% recovery of arsenic 5 as arsenic 3 in this experiment is because of the presence of sulfide in these microcosms, and we get some precipitation of the arsenic 3 that's being produced as arsenic 3 sulfide minerals. So this is showing just the aqueous concentrations. But you can see that we do have arsenic 5 going away with time in the incubations, and we see arsenic 3 being produced in live conditions. And if we give them some different possible electron donors here, we see that this activity is stimulated by addition of lactate as an organic carbon source. But it's most especially, in the case of these sediments, most especially stimulated by amendment with the inorganic electron, autotrophic electron donors, sulfide and hydrogen. Okay. In a subsequent paper, we looked at the effects of salinity on arsenate reduction in the Searles Lake sediment. Okay, so in this case, I just set up microcosms on a salinity gradient, going from 50 grams per liter up to salt saturation at 346 grams per liter. And as we saw in the last experiment, we do have microbial reduction of arsenic 5 at salt saturated conditions in the Searles Lake community. But this experiment shows that actually the overall community in Searles Lake seems to prefer less salty conditions, and that there is certainly a component of the community that is most happy reducing arsenic at low salt concentrations.
Still seawater. Yeah, right.
Right. Yeah. Right. Good point. Still above seawater. And if you kind of think about it, there are wet seasons at Searles Lake when in the spring when the meltwater is coming in most heavily, where salinity concentrations do drop down, and you have lenses of fresh water that can be established sitting on top of the more dense water. So these bugs do see lower salt concentrations sometimes. We also looked at salinity effects on sulfate reduction in sediments from both of these lakes. And one kind of interesting finding we found was that in the case of Mono Lake, which is of these two, the lower salt lake, okay, but about 90 grams per liter, 80 or 90 grams per liter. And if we look at sulfate reduction rates in the sediments from Mono Lake, we see that sulfate reduction does drop off as you increase your salinity, and that sulfate reduction in the case of the Mono Lake sediments is completely excluded as you approach salt saturation conditions. We were a little bit surprised to find that in the Searles Lake sediments, we were unable to elicit sulfate reduction from that microbial community even at low salt concentrations. So it seems like either the salt concentrations, or as it turns out, it could have something to do with high borate concentrations in Searles Lake. Seems to be more or less completely excluding sulfate reducers from the sediments there. Either they can't tolerate the high salinities or the high borate concentrations. So at the time, we proposed that the reason for the exclusion of sulfate reduction in Searles Lake compared to Mono Lake, while arsenate reduction is present in both sites, has to do with the fact that they can get more energy. The microbes can get more energy using arsenate as an electron acceptor than they can compared to sulfate reducers. So Aaron Oren actually proposed this back in 1999, that the reason that we see sulfate reduction being greatly diminished in a lot of hypersaline settings is because the metabolic energy that's required for halophiles to maintain the cell's internal osmotic salt gradient the energy that's required to do that is too great for the amount of energy that a sulfate reducer gets by using sulfate as an oxidant. Okay, so basically, if you look at the delta G of reaction in this example, where hydrogen is the reductant, using sulfate as the oxidant gives a delta G of -4, and using arsenate gives a delta G for that reaction of -23. Okay, so we propose that basically they don't get enough bang for their buck using sulfate reduction to live in these extreme conditions, but arsenate reducers are able to exist at high salinity. So, you know, if you think about it, this could have implications for the search of life on other planets, looking for life in high salinity brines, for example, in Mars, Europa, or it could have implications, as I'll talk about in a second, for studies of the evolution of primordial life on early Earth. We isolated an organism from Searles Lake that we called strain SLAS1. We later renamed it Haloarsenobacterium silvermanii. And this is a arsenate-reducing bacteria that, in this case, is grown using sulfide as the electron donor. So it is oxidizing sulfide, and it is reducing arsenate and producing arsenite. And it is coupling these reactions to the growth of cells. Okay, so this is an increase in the cell population. If you take away arsenic, it doesn't grow. If you take away the sulfide, it doesn't grow. And as it turns out, this organism is an obligate halophile, which means that it only grows at salinity concentrations greater than about 170 grams per liter. There's no growth at lower salt concentrations for this organism. And it grows best at pH conditions about 9.5 or so, 9.4, which is pretty close to the pH conditions that we see in the lake, which are about 9.8. Okay, so we published this in Science in 2005, you know, showing that arsenic can drive microbial life. It can, you know, provide energy for microbial life at salt-saturated conditions. Okay, and then let me just shift gears a little bit now and take you out of the lake sediments. And what we're going to do now is we're going to go out here to this volcanic island in the middle of Mona Lake. This is called Peoha Island. And it's a little, it's basically a little volcano that's out there in the middle of the lake. And along the shoreline of this island are numerous hot springs that display a wide range of temperatures between 30 and 95 degrees C. Okay, so let's take a look at that. We took a boat out here to this little alcove, and we're going to get out here and look at the shoreline. Okay, so it's a very rough kind of place to work. You know, these are lava flows. The ground's very uneven, and it's sort of an otherworldly place because you've got these very striking hot springs with very striking microbial mats growing of red and green color. The ones with the red microbial mats are about 45 degrees C. The ones with the green microbial mats are about 70 degrees C. And also, I should mention that when you're kind of working over these things, you can't kind of keep your face over them for too long because there's all kinds of methane and ammonia and sulfide and sort of noxious reducing gases coming out here that will make you dizzy very quickly. But this is a reducing setting that's full of those, a lot of those reduced you know, constituents that I just mentioned, and in addition, very full of arsenic-free arsenite. So if we look at what these microbial mat communities are composed of, the green one is a cyanobacteria-dominated mat, oscillatoria, and the red one is dominated by this purple sulfur bacteria, Ectothyroidospora. Okay, so and then also just real quick, let's take a look at the chemistry of the two springs. Like I said, temperature is about 45 in the red, about 70 in the green. Salinity is about a third of that of the lake water itself, because this hydrothermal system involves the ground, not only the lake water, but the lake water is mixing in the hydrothermal system with basically groundwater of the island, okay, the freshwater aquifer of the island itself. But these waters are mixing and they're moving through this hydrothermal system and they're becoming heated, and we have lots of reducing constituents coming up out of the volcanic system. Dissolved oxygen is low in both cases, pH around 9, And we can see that there's plenty of geogenic arsenic here, and that a significant component of that in both cases is present as the reduced form arsenite, arsenic 3. Also lots of reduced things like sulfide, ammonia, methane, like I mentioned before. So we knew that this is a purple sulfur bacterium. And purple sulfur bacterium make a living through the process of anoxygenic photosynthesis. Okay, so for those of you who maybe, you know, if it's been a while since your biology classes, just a quick reminder that anoxygenic photosynthesis is an older form of photosynthesis than the type of photosynthesis we usually think about when we think about green plants and cyanobacteria and things like that. Anoxygenic phototrophs have only one photosystem. They don't have the ability to split the water molecule and to use hydrogen as a reductant to drive the production of NADH and NADPH. What that means is that anoxygenic phototrophs are restricted to living in environments where there is some reduced chemical species in the environment that they can use as a reductant. And if we want to kind of really sort of simplify things, the reductant to fix CO2 into organic carbon. In the case of oxygenic photosynthesis that's used by green plants can live anywhere that they have light and water. Because they take the water, they split the molecule, they use hydrogen as a reductant to fix CO2, and in the process, they produce oxygen that we all breathe. The older form of photosynthesis that evolved before this lacks the part of the photosystem that drives the splitting of the water molecule. So in order to reduce CO2 to organic carbon, they need some reduced species in the environment that they can use as a reductant. And in the process, instead of producing oxygen, they're just going to produce an oxidized form of whatever it is that they're using as the electron donor. So it's been known for a long time that sulfide can drive anoxygenic photosynthesis, that anoxygenic phototrophs can use sulfide as the reductant, and in the process, they produce either elemental sulfur or sulfate. In '93, it was shown that ferrous iron could also serve as a reductant for anoxygenic photosynthesis. 2007, Griffin et al. Showed that nitrite oxidation to nitrate could also drive this process. And in 2008, we did experiments with these purple sulfur bacteria from Mono Lake that showed that arsenite can also serve as an electron donor to drive anoxygenic photosynthesis. Okay, so we published this in Science in 2008. This is some light-dark experiments where we're going to incubate in this case, the red mat material here, both in the light and in the dark, under anoxic conditions, giving them either sulfide or arsenic 3 as the electron donor. So what we see is that in the light, both with the green mat and with the red mat, we see that arsenite is oxidized to arsenate when they're incubated in the light, but in the dark, nothing happens. Likewise, with the red mat, same thing, except with the red mat, it was even faster. So we focused on the red mat for the rest of this experiment. Coincidentally, we also published a subsequent paper that showed you can kind of play games with this. Turn the light on, you have oxidation of arsenic 3 to arsenic 5. And then when you turn the light off, arsenate reducers in the mat community kick in. And then you have arsenic 5 getting reduced to arsenic 3 in the dark. Okay, so you have a light-dark cycle where the overall speciation of arsenic in the tubes goes from arsenic 5 in the light to arsenic 3 in the dark. Okay, so we have a complete biogeochemical arsenic cycle that can take place in this mat community. We isolated an organism that we called strain H.HS1 that grows on arsenic 3 in the light. This is from the red mat. So this is the Ectothiorodiospora isolate. You can see that we're spiking in arsenic 3 here in the light, and they're using that up, and they're producing arsenic 5 in the process. They're oxidizing it. And this is driving the growth of cells. The more arsenic you give them, the more arsenic 3 you give them, the denser it grows. And there's no growth and nothing happens in the dark. We also did radio-labeled bicarb experiments that show arsenic-dependent fixation of bicarbonate. So again, that shows that this is an autotrophic process linked to growth. Okay, so then at the time, we proposed that the real significance of this finding has to do with the potential now to generate the oxidized form of arsenic, arsenic 5, in anoxic settings, strongly reducing settings, in the absence of stronger oxidants like O2 or nitrate. Okay, without these stronger, before the discovery that that photosynthetic bacteria and light energy can catalyze the oxidation of arsenic 3 through photosynthesis, the only way that we knew of to get arsenic 5 was basically by oxidizing it with some stronger oxidant, right? Well, now we've shown that you don't need stronger oxidants in the environment. You can have arsenic 3 oxidation to arsenic 5 based on photosynthesis. You set up the possibility for a complete biogeochemical arsenic cycle where phototrophs can oxidize the arsenic 3 to arsenic 5, and then either autotrophic or heterotrophic arsenate reducers can drive the reverse reaction back to arsenic 3. So at the time, we proposed that this could have implications for early Earth, on anoxic early Earth during the Archean, before the oxygenation of the Earth's atmosphere, before there were stronger oxidants around. And back when, as I was saying, there's reason to believe that there was more arsenic available at the crust of the Earth than there is today. Coupled with the idea that phylogenetic studies of the ARRA gene, the AIO gene, they show that these are very widely distributed. There's a lot of sequence diversity in these genes among the different groups where they're present, implying that this is probably not so much a case of horizontal gene transfer, but that these genes actually seem to be very ancient genes that possibly may have evolved before the last common universal ancestor. So we were pretty excited about 2 or 3 months ago. And this just kind of summarizes the idea that photosynthetic arsenic bacteria may have been active during the early Archean as a possible metabolic source for life prior to the oxygenation of the Earth's atmosphere. So we were pretty excited a few months ago when I was sent this paper to review by this group at the University of Paris, Seforna et al. And this appears in Nature Geoscience now. It was published in November or so of 2014. And what they did was they drilled into a very well-preserved 2.7 billion year old stromatolite in Western Australia, Tumbiana stromatolites. And what they found was arsenic-rich layers in the stromatolite associated with, in some cases, iron minerals, as you might expect, but in some cases associated primarily with organic carbon-rich layers, metalliferous organic carbon-rich layers that are interpreted to be fossilized EPS, fossilized extracellular polymeric substances. In other words, fossilized microbial mat material and basically the slime that microbes secrete in microbial mats. So this shows XRF mapping of the stromatolite fabric. And down here we can see arsenic-rich layers that are associated with organic carbon-rich layers in the stromatolite here. They used Raman spectroscopy to kind of zoom in a little bit more on those organic-rich layers. And in some cases, they're finding what are interpreted to be fossilized microbes. Okay, these are organic-rich globules of organic carbon that's rich in aliphatic, aromatic hydrocarbons, microcrystalline aragonite, and they're analogous to what we see in modern stromatolites when microbes get trapped in the stromatolite during its mineralization. And in this case, we have arsenic associated with these organic-rich fossilized microbes in the stromatolite at high concentrations, associating with those organic-rich globules to the exclusion of the surrounding matrix. This is showing, in some cases, yeah, in the matrix, you've got arsenic associated with iron minerals. But in the case of those globules, it's all arsenic, very little iron or other metals present, except for some copper. That's also present in the organic rich globules. So Saffornia et al. Are proposing that this is sort of the first geologic evidence, the first paleontological evidence, if you will, of microbial arsenic cycling 2.7 billion years ago on primordial Earth. Basically, that's the dawn of life. So it would be nice, one thing that would be nice is if we could do some stable isotope studies, right? Because when microbes cycle metals or when they cycle sulfur, for example, or iron, you might expect to see a stable isotopic fractionation that's associated with biological processes. And it would be nice if we could look for, to see if there's a stable isotope effect here, but unfortunately arsenic has only one stable isotope. So that brings us to the next part of my talk, which is the related element, antimony, which does have more than one stable isotope. And I'd just like to mention that because ultimately I think it would be interesting to start to look for antimony-rich deposits in the geologic record and see if we can find evidence of any kind of a isotopic fractionation. But that's still further down the road because we're only starting to really understand how microbes cycle antimony. So in order to study antimony cycling in the modern, I went to this mine site that I mentioned in Idaho, the Stibnite mine. Okay, it's a little tough to see, but this is the border of Idaho here on this map. And this is where Stibnite mine is located in the center of Idaho in the Payette National Forest. Okay, this is a gold and antimony mine that operated from the 20s through the 90s. And now there's exploration going on to reopen it because the price of gold has come up sufficiently that mining companies are interested in getting back in there. But it's currently listed as an EPA National Priorities List site because over the course of the mine's operation, massive mine tailings piles have been piled up sort of in the stream valley here near the mine that are releasing high concentrations of arsenic and antimony into this creek, Meadow Creek, which is a tributary to the East Fork Salmon River and is a spawning area used by endangered Pacific salmon, okay, among other problems here. So this is one of the mine tailings, and this is a truck on top for scale, just so you can see sort of how big these mine tailings piles are. And they're leaching all kinds of arsenic and antimony into the environment. There have been some remediation efforts to kind of lower metalloid concentrations in Meadow Creek. The creek has been rechanneled so that it flows around the tailings, and they've installed some sand filters. And it seems that these things have, these strategies have helped in bringing down arsenic and antimony concentrations in the creek compared to pre-remediation values. But where we do continue to see exceedingly high aqueous concentrations and sedimentary concentrations of arsenic and antimony are in seeps and springs and sort of wetlands that occur sort of just on the periphery of the tailings piles themselves. So I'm going to show you some data from experiments using sediments from some of the more contaminated seep and spring pond sites here at this mine. The most contaminated site we call heap seep, which is next to that large tailings pile I just showed you. And we have arsenic concentrations there in the water up to tens of parts per million. Okay, this is dissolved arsenic. And antimony concentrations of up to 1 ppm or so. And then some other highly contaminated ponds where we did some microbial culturing. Okay, so very high arsenic concentrations. Here is the heap seep. This particular picture was taken during a field visit when it had been kind of dry. Okay, so this is not a permanent wetland, but it is generally contains some amount of water because this is a low-lying spot. This is actually where they had the leachate collection point for cyanide leaching during the 80s. when they were doing cyanide leaching to try to leach gold out of the mine tailings pit. And this is where they would have collected the leachate. So we get lots of water that kind of accumulates in this low-lying spot. It's really nasty. At the time, it was so evaporated that the concentration was actually 20 ppm or so. pH was very high, I think because of the cyanide, and antimony, again, around 1 ppm. There's nothing that really lives in this little wetland except for a thick microbial mat that's probably photosynthetically dominated, and then boreal toad tadpoles that are feeding off of the mat, okay, living at these incredibly high arsenic and antimony concentrations, high pH, and eating incredibly contaminated mat material. And this place is, I'll show you some pictures in a minute, this thing is just teeming with tadpoles in some places, of the boreal toad tadpoles. Okay, so I'll talk about the tadpoles again in just a minute, but let's take a look at some geomicrobiology here for the antimony. So I published a paper in ES&T last year that shows microbiological reduction of antimony 5 and also arsenic 5 in sediment microcosms from stibnite mine. Okay, so again, setting up anaerobic microcosms with sediment from the site in an artificial media that's designed to mimic freshwater chemistry, giving them arsenic or antimony, in this case giving them lactate as an organic substrate. And we were able to elicit, well, first of all, let's take a look here, 2 millimolar arsenate. So this is arsenate reduction in anaerobic microcosms of the sediment. Okay, arsenic 5 being reduced to arsenic 3, nothing happening in killed controls. So that shows this is a biological process of arsenic 5 reduction. And when we give them 3 millimolar antimony 5, we also see in the live microcosms a loss of antimony 5 and nothing happening in the killed controls. Now we would like to see recovery as antimony 3 here, and we're not seeing it in these particular microcosms. I'll show you why in just a minute. But let's take a look here. When we give them both metalloids concurrently, we see that this population of organisms will use arsenic 5 first, and then when the arsenic 5 has been depleted, the antimony reducers kick in, and we see a loss of antimony 5 at that point. Okay, so why aren't we getting good recovery here? And it has to do, again, with sort of a similar situation I mentioned a little bit earlier with the mono lake slurries. We're getting a precipitate. And so this is now as an enrichment culture. This is an enrichment culture of antimony 5 reducing microbes that we're enriching from the sediment microcosms. We're diluting them away from the sediment. We're growing just the bugs now in the media, amended with, in this case, antimony 5 and lactate. Okay, the lactate's an electron donor. It's going to drive the reduction of antimony 5 to antimony 3. And as it turns out, in these particular experiments, we were using a media recipe that contained sulfate. So in addition to antimony 5 reduction, we also had sulfate reduction going on here. And when the antimony 5 reduction occurred in the presence of sulfide, we end up getting this bright orange precipitate, this mineral, which is the mineral stibnite Sb2S3. So this is an antimony 3 sulfide mineral. And this is biological precipitation of the stibnite. So the tube goes from this clear color to being full of this bright orange amorphous stibnite mineral as a result of the microbial reduction. So we did Zane's analysis because this was amorphous and we couldn't just run it on an XRD. So I took it over to Stanford synchrotron and we ran it comparison to some different antimony standards. In this case, this is abiotically precipitated stibnite, and we can see that there's a good match with the precipitate that we're getting from the culture here. So when the computer kind of works up the data, we find, like, for example, here in the enrichment culture, this precipitate is 96% antimony 3 sulfide. Okay, so that's where the antimony 3 is going during the antimony reduction. We showed that this was a respiratory process in the microbial community here, not just a detox, but that the reason that the microbes are reducing the antimony 5 is a respiratory process. They're using this to get energy. This is a metabolic process. So in order to prove that, we gave them radiolabeled acetate as their organic carbon source, and we show the production of radiolabeled CO2, antimony-dependent oxidation of 14C acetate to CO2. So in other words, the more antimony 5 we give them, the more they oxidize this organic carbon source to CO2. Okay, so they're coupling the oxidation of acetate with the reduction of antimony, and so this is a respiratory process. It's not just a detox. Around the same time that we published this, Tim Hollabaugh's group at University of Georgia actually isolated an organism that grows this way, and so his paper showing an actual growth of an isolate respiring antimony 5 to antimony 3 appears in the same issue of ES&T. So rather than show you Tim's data for that paper, I thought I'd just show you a little bit of new data from an organism that we isolated from these stibnite mine sediments in my lab. And so this is some data that my current grad student, Megan Dovick, produced from her isolate that we call strain ID SBR1. This is an arsenic, in this case, an arsenic 5 reducing, but also antimony reducing, as you'll see in the next slide, organism of the genus Clostridium. Okay, so here she's spiking in arsenic 3. Okay, she's spiking in arsenic 3. It's using the amendments. It's producing arsenic 5, and it's coupling this to the growth of cells. Okay, and this is heterotrophic. We're giving it lactate as the reductant. And we see a similar sort of thing going on when we give it antimony 5. Antimony 5 is being used and is driving the growth of cells. If you don't give them antimony, they don't grow. But we don't have good recovery again. So now where is the antimony 5 going? Because we're using sulfate-free media this time. We're trying to avoid that problem of the precipitation of the antimony of the stibnite. Well, so as it turns out, we're getting precipitation of another mineral. Antimony compounds are much less soluble, generally speaking, than arsenic compounds. And so when microbes, what we're finding, and what other people are finding more and more, is that as microbes cycle antimony, it's a lot more likely to result in the precipitation of the antimony as some insoluble solid form. That's what's going on here. As her bug is reducing antimony 5 to antimony 3, we're getting precipitation of antimony 3 bearing mineral phases. And these we could measure on the XRD. So this is showing XRD traces of this mixture. And we see that, yeah, okay, she's putting it in as antimony 5. We do get some precipitation of the antimony 5 mineral mopungite, but we also get considerable precipitation of the antimony 3 bearing mineral synarmentite. So this is an antimony 3 oxide, and this is where the antimony 3 is ending up as the microbes are reducing antimony 5 to antimony 3. Okay, so this is precipitation of a mixture of antimony 5, antimony 3 bearing mineral here. Currently now, we're working with another organism that appears to be growing as an autotrophic oxidizer of antimony 3 and also arsenic 3. We call this strain ID SBO4. This is a different strain than the last one. And this organism is the genus Variovorax. And this organism is going to oxidize antimony 3 to antimony 5, and couple that to the growth of cells. And also it can do the same thing with arsenic. We have done radiotracer studies with radiolabeled bicarbonate that show antimony 3 dependent fixation of radiolabeled CO2 during sp3 oxidation, which suggests that this is an autotrophic process and that it's linked to growth. The reason that I say we're not 100% sure is because these experiments were done using antimony in the form of an antimony 3 tartrate. And so we're trying, you know, because of solubility issues, we're trying to work with an antimony source that doesn't contain tartrate, that's basically, you know, organic free. But we have strong evidence so far to support the idea that this is autotrophic oxidation of antimony 3. In other words, they're coupling this to the fixation of CO2 and to metabolic growth. Okay, so this is still a work in progress. But this is, you'll notice we get, you know, decent recovery of antimony 3, but not complete recovery. And my student who is growing this bug again has shown that some amount of the antimony 5 that's being produced in this case is ending up as the mineral mopungite, okay, as an insoluble precipitate during microbial cycling. Okay, so we see lots of precipitation, both oxidation and reduction of antimony 5 and 3, coupled to autotrophy, coupled to heterotrophy, driving the precipitation of either stibnite mineral or mopungite or synarmentite, primarily. So that brings us back to the tadpoles, okay? If you look at the microbial mat that they're eating here, it's kind of an odd color, kind of this orangish tan color. And as it turns out, this is a very sort of gelatinous, soupy kind of microbial mat. If you put your hand in there, it's really squishy and it's several inches thick. It's a very luxuriant microbial mat. But it is full of flocculated arsenic and antimony sulfides and iron oxyhydroxides that have tons and tons of arsenic and antimony associated with them. So the microbial mat has arsenic concentrations 89,000 ppm. Okay, and the reason is that it's just full of basically arsenic sulfide minerals. And then here's the antimony concentration down here for that microbial mat, 2,000 ppm. And if we look at the tadpoles that are eating the mats, incredibly high whole body concentrations in the tadpoles. In fact, to our knowledge, these are the highest concentrations in living tadpoles that have ever been reported. This is how dense the tadpoles are in some parts of that heap seep. Okay, they just teem in there. And so my student Megan is working on a project looking at the survivorship of the tadpoles as they mature, and also looking at the concentrations of the arsenic and antimony in the tadpoles as they go through different maturation stages. So here's some interesting data that she produced. In this case for arsenic, looking at whole body arsenic concentration as it changes with metamorphic stage. And when we saw this, this was kind of, kind of our first indication of what was going on with these high arsenic and antimony concentrations. Because when the tadpoles reach this stage right here, where the arsenic concentrations drop off, this is the stage where they're absorbing their tail, they're growing their limbs, they're getting ready to turn into a toadlet instead of a tadpole. And at this stage, they stop eating. They stop eating here. And they basically excrete everything that's in their gut. Their gut goes through radical metamorphosis. So obviously we thought, it could have something to do with what they're eating, and we tried to purge the tadpoles before we analyzed them, using sort of standard methods where you hold them in water for a few days. You try to let them excrete all of the stuff that's in their gut, so that you're not measuring the gut content when you actually measure the tadpole. So using standard methods, we tried to purge them. We still got these incredibly high concentrations, but then we saw it dropped off here, and we said, something must be going on with the food. So when Megan opened up the tadpoles, even though they had been purged, this is what she found. So this is the tadpole gut, and you can see it's just packed full of this orange stuff, which is the mat material. Purging was not effective in this case. But when we put this stuff on the electron microprobe, arsenic sulfides, antimony sulfides, and iron oxyhydroxides associated with high concentrations of arsenic and antimony. Basically, it's the same thing that we see in the mat. So what we're proposing is that these flocculated and in some cases amorphous minerals that are being precipitated in the mat are the direct products of microbial antimony and arsenic cycling that's driving the precipitation of these minerals in the microbial mat. And then that in turn is a source of high concentrations of arsenic and antimony to the tadpoles, to that part of the food web through ingestion. Okay, and she also did some other work looking at concentrations in insects and plants and things like that. And if you'd like to know more, I'd be happy to tell you about it, about her study. Okay, so how do they pass it? You know, we're not sure why the purging was so ineffective. To be honest, Megan thinks that they were just eating their waste in the purging, you know, and they're just like kind of re-eating it. But the biologists, we're collaborating with some USGS biologists in Idaho on this study, and they're just, you know, obviously amazed by the concentrations, amazed by the fact that purging was so ineffective. But another interesting thing about it is that a lot of times people think of amphibians as being sort of the indicator species, sort of like the canary in the coal mine, and that they're one of the most susceptible, you know, organisms for a lot of things. This study shows that, at least in the case of the boreal toad tadpoles, they're much more resistant to metal toxicity than we previously
Other
mentioned.
than the gut.
Yeah.
Where's the arsenic?
Okay.
Where is it distributed?
She didn't break down the individual organs, but about 90%, roughly 90% of the arsenic and antimony concentrations can be accounted for just from the gut content. The rest, you know, is just, she basically did three fractions. She did the gut, she did the tail, which is sort of just the muscle, and then she did the body cavity as a whole, which would kind of include the other organs. Out of that, like I said, about 90% in the gut. The bulk of the rest was in just the body cavity, and very little in the tail muscle itself. Okay.
They do become frogs. So.
Oh yeah, and they do become frogs. Yeah, that's a good point. Yeah. Yeah, that's a good point. And I wasn't sure how much time I would have, but she did do she's working on a survivorship study, and actually she shows that the rate of growth for these boreal toad tadpoles is pretty typical of boreal toad tadpoles, even in non-contaminated sites. So they're growing just as fast, they're maturing just as fast. There's no obvious evidence of deformation of the tadpoles when you go out there and look at them. And at the end of the spring, they all turn into little toads and they all leave the pond. And whether they are fertile and whether they can go on to reproduce, we don't know, but we do know that they make it to maturity. And that, you know, at least anecdotally, there's no evidence of lots of deformations or anything like that in the population. Well, that's a good. Well, I wouldn't be so sure because birds don't know. You know, birds don't know that they're high antimony, so it could be moving into other parts of the food web through the tadpoles. You know, that's another question. Okay, so oxidative, reductive, and transformations of arsenic oxyanions drive a wide range of microbial metabolisms, and that they may have been active on primordial Earth. And like arsenic, redox transformations of antimony oxyanions can be harnessed by bacteria to obtain metabolic energy. This is a relatively new finding. You know, just in the past few years, have we realized this? Well, really, you know, in the past year or so, really we're making, you know, a lot of progress here right now. Separate bacterial isolates from stibnite mine grow by heterotrophic respiration of antimony 5 and possibly autotrophic antimony 3 oxidation, and the cultures also grow using arsenic. We're working, and other groups are working, to try to figure out whether antimony and arsenic oxidation and reduction are encoded by the same functional genes. In other words, is it those same arsenic genes that I mentioned earlier that are driving these transformations of antimony, or are they different enzymes? And it's looking like they are primarily mediated by distinct and different operons and different genetic operons and different enzymes. Microbial transformations are interpreted to be a critical component of the global biogeochemical antimony cycle and to exert a profound influence on the element's bioavailability and environmental behavior. Okay, so with that, I want to thank my collaborators, and these are my two grad students who produced some of the data that you saw today. And these are the folks at USGS, both with Ron Ormland's group and also the two fellows in Boise, Idaho, who are USGS biologists working with me here at the Sediment site, and then Heather Wyatrowski at Clark University, who works with me with some of the molecular biology end of this. So thanks very much, and I really appreciate the opportunity to talk today. Thank you.
Thank you for a fascinating and wide-ranging presentation.
Yes.
Great questions for Paul. So the star of the show here. That's mature.
That's a toadlet. Yeah, that's a young one just coming out of the pond. Yeah.
So these are in the heat.
Right.
So the mining activity is only 100 years old.
Right.
So are there other naturally occurring ponds?
Sort of deposits or natural.
Yeah. Are there sort of homologs or analogs, whatever the word is, of the heat seals? So these tadpoles would have seen this environment before.
Yeah, I mean, it's hard to say it's hard to say. First of all, it would be interesting to try to expose a population of boreal toads from a pristine site, to see if these are genetically predisposed in some way to be more tolerant because of the fact that they live around the mine. There's not a lot of naturally high antimony places in the United States. Stibnite Mine is one of the few. It was a strategic reserve for antimony during the World Wars because it's used as an alloy in ammunition. And it's one of the few places in the United States where we have a commercial deposit of antimony. This is a gold mine, but also an antimony mine. Arsenic and antimony often co-occur with hydrothermal ore deposits because they are chalcophilic elements. They're sulfur associated, and they tend to be associated with hydrothermal settings. But in terms of high arsenic concentrations, we have this in the United States. We have some places in Nevada that are also associated with hydrothermal settings. There's some high antimony, for example, around Yellowstone. The place where antimony contamination is really a big problem right now is in China and in parts of Australia. There's large antimony mines in China, one in particular, where most of the world's antimony supply is coming from right now. And it's increasingly being used more and more in industry as an alloy and in electronics. So they're producing massive amounts of antimony, and it's producing massive antimony contamination. And there's a lot of people in China, a lot of scientists in China, who are starting to study that site.
So in terms of Superfund sites, is this unique with regard to antimony?
I think so, as far as I know, unless maybe there's some industrial, places. But as far as mining, I think it is. Yeah.
So you pointed out that the crustal abundance of antimony is an order of magnitude below that of arsenic.
Right.
Is there any evidence that if you have 1,000 ppb groundwater somewhere, that you might have 100 ppb antimony in it? How much has been published about antimony in reducing groundwater?
I've found very, very little about antimony in groundwater at all. Under reducing conditions, it might not be terribly soluble, as we've kind of been talking about. And certainly if you have sulfide present, it's probably going to not be very soluble.
But you wouldn't. In the Bangladesh aquifer, you wouldn't find, sulfide.
Right. Okay.
So have Bangladesh well water samples been looked at? So we did early on when we were doing the method development, and we didn't see anything, huge, but we were not targeting the super high arsenic samples, and we might need to revisit that. I think the WHO guideline for antimony is similar to arsenic. I think it's 10 ppb. I remember looking that up at some point. So from the toxicity point of view. Other questions for Tom? Well, thank you again.
Thanks so much. I enjoyed it. Thank you.
You could see, but people were signing off.